So these macrophages, they're really cool cell types, and they were discovered over 100 years ago. But there's still so much we don't know, and there's so much debate about how many different functions they have and how we can actually classify them. So a few decades ago, scientists agreed that macrophages can exist in two different extremes. One is where they encounter an enemy, right? A bacteria or a virus, and they need to respond to them right away. So they do this by sending out chemical signals of inflammation, which tells other cells in the body that something is going on here and they need to check it out right away. And this is a great thing. It tells the body that something is wrong, and it's very necessary. But too much inflammation is not a good thing because it not only damages the bacteria or the viruses, but our body itself, and that's not good. So macrophages are so good that they can actually switch their function when they recognize this, and they become anti-inflammatory, which is the other extreme of their existence, where they're trying to carry out repair functions and trying to heal wounds by releasing proteins and by building new blood vessels. So scientists were satisfied with this explanation and this definition of macrophages being at the two different extremes for a few years. But then as we started carrying out experiments, we realized that the cells are actually a lot more diverse and a lot more plastic than this. And they exist anywhere in between these two spectrums, between these two extreme states, and they can actually be in any state in between this, which is influenced by something in their environment. And they can actually switch rapidly between the different states that they exist in. And just think about it, it's mind-blowing. So if the body actually needs to heal a wound, then the macrophages can become wound healing cells because of the signals that they get. And if they need to launch a massive attack against a bacteria or a virus, then the macrophages will actually cause the inflammation that's needed to defeat the virus. So what was earlier known was that chemicals were responsible for the macrophages making the switch, right? So chemical signals from the bacteria or the viruses or the healing wounds will actually interact with the macrophages and tell them what they need to do. But what is being evident in the last 2 decades, and what I was doing as part of my research, is that we can actually influence these macrophages with the help of physical cues as well. So something as simple as how soft a material is or how stiff it is can actually influence how the macrophages are. So for example, if you take a soft biomaterial like a contact lens, right, it's going to have a different response compared to a stiff material like our hip implants. So it's not just the stiffness, right? A lot of different cell types actually do respond to different physical forces like shape and the size and the stiffness or the elasticity. But the extent to which the macrophages actually change their behavior is fascinating in this respect. So I found, for example, that based on just the material they're sitting on, if they're sitting on a really stiff material, as stiff as our bones, then the cells are going to go into an inflammatory state, right? Whereas if they're sitting on really soft materials, like our skin or our cartilage, which is found in the ears, then this makes the cells into a much more wound healing phenotype. And it's not just the stiffness, right? It's the shape of objects, it's the size of objects, it's the kind of cues that they're presenting to the cells and what we can do with it. So think of it this way. What if we're delivering nanoparticles to our body and we're delivering drugs through these nanoparticles? Can we change the shape of these nanoparticles so that we can make the macrophages respond the way we want? For example, needle-shaped particles pierce through the cells, right? The cells are very delicate. So when the needle-shaped particles pierce through the cells, they cause more of an inflammatory response compared to spherical particles, for example, which don't pierce through the cell. And if they're small enough, they're capable of being taken up by the cell. So, and it's not just implications in inflammation. These physical parameters actually help a lot in the wound healing process in whatever part of the body they are in as well. So they have a really important role in a lot of regenerative processes in our body. And my research actually showed that it was important to have both the extreme types of macrophages, both the inflammatory type and the anti-inflammatory type, in order for bones to heal properly. And if there was only one or another type, then there wasn't as much new bone as when there was a mix of the two different extremes. So finding out more and more about how these cells respond to the physical factors as well can have a huge effect on how we design the biomaterials that go into our body. So if you can think about it, do we coat hip implants with softer gel-like materials so that we can reduce the inflammatory response after they're implanted? Can we avoid sharp edges on materials and make them rounded instead? These are some of the design materials, design parameters that are being studied today in terms of biomaterial design. And it's really cool that macrophages are actually informing this design. So where in the body do we find macrophages? That's another cool thing about these cells, is that they're everywhere in our body, and they've evolved so much in each part of our body that they have different names and completely different functions depending on where they are. For example, macrophages in our bone are called osteal macrophages or osteoclasts, and they help in the constant turnover of bone and help to repair small and large injuries as well within the bone. And macrophages in the brain are called microglia, and they're quite cute to look at. You know, they look like they've been splat on a flat surface, and you're going to hear a lot more about them from Professor Marina Lynch later today. So macrophages are also constantly circulating in our blood, and they're looking out for infections and any sign of distress so that they can make their way to wherever they need to be very quickly. And they really are everywhere in our body. Just imagine if you get a paper cut, by the time you realize that you're hurt, macrophages are already there trying to heal it. So let's go back to tattoos, right? And let's see if I can convince you to get a tattoo. So what happens when we get a tattoo? The ink is injected into the second layer of the skin, which is the dermis, and that's below the outer layer of the skin, which is the epidermis. And the reason we don't just inject the tattoos into the epidermis is because it has a very high turnover and the tattoos would disappear very soon. So the tattoos go into the dermis, which is under the first layer of skin. And what happens here is the ink particles are chemicals, right? So the particles are kind of just suspended under the skin, and the macrophages are hungry. They want to eat anything and everything. So they go and they try and eat these ink particles. But the chemical inks are not actually like bacteria or viruses. So the macrophages can't actually get rid of them. So they just kind of hold on to them and float around there